Everyone. My name is Gizem Taş. I'm a PhD student at Tilburg University, and here I wish to present our work on nonlinear dimensionality reduction of genotype data using autoencoders. Our valuable genotype data comes from Project Mine. Project Mine is a study that is all about collecting genetic data from ALS patients and control subjects, and mining and comparing that data to find the genetic cause of the disease. The ultimate goal is to find the concrete starting point for new treatments for ALS. Let me first establish the fundamental problem in the big picture in our genetic ALS research. Linear methods like GWAS are unable to capture epistatic interactions between single nucleotide polymorphisms. Deep learning is highly promising for uncovering these complex genetic architecture of ALS. The feature space of the Project Mine data is too large for off-the-shelf use in deep learning. Therefore, nonlinear compression is required. For instance, our current sample includes over 110,000 individuals with over 6.5 million of SNPs as features. Therefore, autoencoders are promising to compress genomic data with minimal information loss. Here comes our pipeline. Our goal is to compress high-dimensional genotyping data while preserving disease-causing genetic patterns. The main challenge is finding the balance between the curse of dimensionality and unwanted loss of SNP information. Our approach entails dividing the genome into non-overlapping and correlated clusters of SNPs, which are called haplotype blocks. Then, building and training autoencoders individually for each haplotype block. Tuning hyperparameters for each model is computationally demanding. Therefore, we optimized hyperparameters only for a subset of 221 haplotype blocks. Autoencoders are self-supervised neural networks which can compress and encode data in a low-dimensional bottleneck while learning to reconstruct the input data. The encoder compresses the input data into the bottleneck layer. Then, the decoder uses this compressed data to reconstruct the input data in the output layer. Haplotype blocks have different characteristics. Therefore, one should not use the same architecture for compressing all haplotype blocks. We adopted an adjustable network structure based on 3 hyperparameters: number of hidden layers, the bottleneck dimension, and the shape of the network, either rectangular or elliptic. The average fitting times of the best models across all cross-validation faults resulting from the grid search show that higher levels of compression require deeper and wider autoencoder architectures. Therefore, if we want to compress the data more, we need to access more computational resources. Then, the average validation reconstruction accuracies of the best models, average over 221 haplotype blocks, along with their confidence intervals, indicates an algo point observed at a bottleneck dimension of 3, beyond which the incremental improvement in accuracy diminishes. The average SNP accuracy improves only marginally with an additional bottleneck dimension between 2 and 3. Then, the relationship between the block size and the SNP accuracy is affected under different scales of compression. On the left, the more rigorous the compression becomes, the less robust the SNP accuracy is to increasing block sizes. Furthermore, the internal genetic variation of the blocks can be quantified using the average pairwise linkage disequilibrium. On the right, we see that the positive correlation between the accuracy and the average pairwise LD gradually becomes weaker as the bottleneck size increases. Finally, using the chosen autoencoder architecture per haplotype block, we compressed the entire chromosome 22, which originally contained around 70,000 SNPs clustered in 2,854 haplotype blocks. This entire chromosome was compressed down to 10.45% of the original input size, with an average SNP accuracy of 99.51% on the training samples and 99.52% on the test samples. Our contribution is therefore threefold. First, we propose an efficient dimensionality reduction approach leveraging the LD structure in the genome and extending beyond linearity using autoencoders. Second, we provide an adjustable autoencoder structure compatible with each individual haplotype block. We use this structure to provide a standardized approach to bypass extensive hyperparameter tuning. Third, we show on an entire chromosome that using autoencoders to compress haplotype blocks can achieve a compression of the data to approximately 10% of the original size with a reconstruction accuracy around 99%. Thank you for listening to my presentation.